Welcome to the class of microarray technology. Dear students, today we are going to learn about supervised learning and unsupervised learning methods that are used to analyze the microarray data. So in the last class you have seen how we obtain the microarray data and how we normalize that and how we transform the microarray data. Now we will discuss what are the different methods that can be used. So supervised learning and unsupervised learning are the two different methods. So first of all we will discuss about supervised learning. So it depends on the annotation or data. So supervised learning always use the annotation and predefined information in the analysis of gene expression data. So it can be classified into the two ways. So in the supervised method we used predefined data. Okay, we use the annotation of the either the gene or the sample, means to which sample it belongs, what is the source of the sample, suppose cancer, non-cancerous, or any disease. It can be any disease also. It can be obtained from the animal, plants, or any specific plants under any circumstances, under any conditions also. So we have the predefined information about the sample data or gene and proteins. Okay, so that is used to cluster of the gene or sample in order to identify the pattern. So previously we have the information and that can be used for the classification. So we can say this is the type of the classification because we previously we have the information. So to cluster the genes we use the predefined information. Okay, so on the basis of that we can also identify the patterns that are similar in that sample. Okay, and that may be the characteristics for the cluster or for the group of the genes. But what is the important? That is the important information. We have the predefined information. We have so for example we can use the separate sample expression profile. So when we are going to analyze the sample expression profiles, so it can be separated into the disease state and normal state groups. So from the same source we can have the sample in the two state, like disease state and normal state. Okay, or we can take the sample at the various stage of the disease, like initial stage, acute stage, and chronic stage. Okay, so there can be the various samples. Then look for patterns that separate the sample profile of the disease state from sample profile of the normal state. Okay, so then we can do the comparison between these two, the normal state and the disease state. So this is the supervised learning method in which we use the predefined information, and that is very helpful in clustering of the genes. So we should say this is the classification method. Like the classification, we make the different class but we have the predefined information, like in the roll number or anything. So it can be used to make the groups, roll number, name. Okay, so this is the same thing which is applied here, and that we call that supervised learning because we have the predefined information. अब बंद ही रखना पापा, फोन मत करो
इसे, नहीं अब कुछ मत चलाओ, बंद रखो इसे, बाद में चलाना, अभी बंद करो।
Now come to the unsupervised learning method. What are these? In the unsupervised learning method, the expression data is analyzed to identify patterns. But we do not have predefined information here. We do not have anything. We do not know about the source and any information about the expression data. So that can group genes or sample into cluster. So without using any predefined annotation or any predefined information. For example, genes with similar expression profile can be used to cluster together without use of the any annotation. Like we have we have the gene expression data, but we do not know about the sample. Means in which condition it is grown, it belong to the specific disease or not. Okay, so any clustering is based on the output of the, what is the output of that from that data. On the basis of that pattern, what expression they are showing, after analysis of the their patterns, then only we can group and cluster together the genes. So annotation is obtained after the analysis. That is only used, that pattern is used for the clustering. Okay, so we can understand this unsupervised learning like in the same class we use the roll number and name for the classification. Suppose we have the different classes like M.Sc Biotechnology, M.Sc Bioinformatics or B.Tech. So these are three different classes, or B.Tech Chemical Engineering, or different classes we have. Or we can take the examples like B.Tech in the specific branch, Chemical Engineering, Electronics and Mechanical Engineering. Okay, and so in the class, okay, in all the session, the students have been assigned the different seminar topic. So when we classify the student on the basis of the roll number, name, and on the basis of their branch, so that is known as, this is the classification method, and that is a supervised learning, because we have the predefined information like chemical engineering students belong to the B.Tech Chemical Engineering, students belong to the B.Tech Electronics, or students belong to the M.Sc Bioinformatics or M.Sc Biotechnology. Okay, now what is the unsupervised learning? Like suppose we have different seminar topics like 20, 22, okay, or 15. It can be anything. Suppose there can be the four seminar topic. Now I told to the student that you choose any one topic from this group of topics. Okay, so students will choose the topic. So according to the output of the data, we can assign the different group like 4 groups or 15 group or 16 groups. How many seminar topics we have? So students can be grouped together on the basis of their choice, what they have chosen from the seminar topics, because we have different categories of topic like application of mechanical engineering in environment, application of electronics in environment, or application of computer science engineering in environment. So on the basis of their choice, or there can be different topics also, so on the basis of choice we can group them together. So that is called clustering, because they have previously I do not know which topic they are going to choose. So I have allowed them to choose any topic from any different branch. Computer science student can also choose topic from the different for according to their interest, because generally the other branches students also choose their
seminar topic that belong to the computer science also. So it depends on their choice. Previously I do not know. I have listed the topic according to their branches, but previously I didn't know about that they will choose the topic other than their branch. So this is called unsupervised learning, because after their choice, when they have mentioned their choice, then I group them together. So this grouping is called unsupervised learning. This is based on the unsupervised learning. It is based on the pattern that they have chosen their seminar topic, same seminar. The students who have chosen the same seminar topic, that is grouped together. Or there can be another criteria also, like the students from the same branch, if they are choosing the same topic, so they can be grouped together. But the first one example, the first in which we are using the first criteria, that is a suitable example of the unsupervised learning. I think this should be clear now. If we have the predefined information annotation, that means it is supervised learning. But if we do not have the predefined annotation or information, that is called unsupervised learning. Okay. So in the case of unsupervised learning also, we can use the annotation at the later stage to make the meaningful prediction. Suppose the data belong to the cancer data, so we can use that information for any confirmation or for any analysis. But what is the idea behind that unsupervised learning? It is purely based on the outcome patterns, the output of the result. What result? Means any predefined information should not impact on the output. Suppose if you already know this belong to the cancer cells, okay, then we are not going to do the any analysis. It impact the result. We already have information. It may impact the result. But if you want to analyze, if you want to give the correct predictions, then unsupervised learning is good. So because it is based on the output of the result, the genes, suppose in the case of gene expression data that are showing the same patterns, that will be grouped together on the basis of their expression, because there is no predefined information in use. Because if we use a predefined information, that is means we are already classifying the data, so that is not the grouping or clustering. So it is purely based on the output. Now after the analysis of the data, we are classifying and grouping the genes on the basis of their pattern that is shared among the data. What is the pattern? Pattern means that is shared among the data, that is common among the data. If they are sharing the common pattern, means that like students are choosing the same topic, they will be grouped together. So that is called clustering. Although we can use the predefined information at the later stage to make the meaningful biological inference, means in case of gene, the genes have the similar expression and they belong to the same disease also. So we can make the meaningful biological inference in case of gene expression data. So gene may belong to the same stage, gene may belong to the disease state, or genes may belong to the normal state. So we can compare what we already have, the information. We can confirm that predictions. Okay. So we can also analyze the genes expression data that is obtained at the various states, normal state, abnormal state. Okay, so today we have discussed about supervised learning and unsupervised learning methods. In the supervised learning method, we use the predefined information, that is the classification. Means we are doing the classification on the basis of predefined information. Like I took the example of the class. In the class we use the name and roll number. So in the case of gene also we use the gene ID, or we can use the source of the gene
also, like cancerous, non-cancerous, plants, animals. So that is called supervised learning method, where we use the predefined information for the classification. In the unsupervised learning, we do not use the predefined information or the sample. So it is based on the output pattern, expression, gene expression data. It is based on the gene expression data. Data which share the common patterns. So expression data is analyzed to identify patterns that can group genes or sample into clusters. Okay, I think now it is clear and we have understood using the suitable examples. Now it is okay. Now the class is over.